Hey everybody, I hope your first day went well. So we're now going to recap what we learned today to try to help you integrate it into a bigger picture modeling framework. So as we've just learned, maybe one of the most intuitive ways to classify models is to think about the types of potential questions that they might answer. So for a given problem or question, we can think of it as fitting into one of these three categories. So the first is this a descriptive category, a descriptive question about what is happening in a system? Or maybe this is a mechanistic question about how a system is actually working. Or finally, this could be kind of an interpretive question about why a system behaves in the particular way that it does. So the models that we built today and the models that we build in general can ask or help us ask some of these broad categories of questions. And as you'll see over the next couple weeks, models typically do fall into these three general categories. Of course, they can span across categories too. They don't always fit nicely into one category only. So today we went over one version of each of these models, a what model, a how model, and a why model. And we did one example of each with the idea that you can make it really concrete. So in today's wrap up, we wanted to share with you some other examples of these types of models. And the way that I'm going to do that today is to share with you how we've been using these types of models in the research that I do in my lab, in my own research. So in the first tutorial, we saw a what or descriptive model. And what this did was examine the spike trains and the interspike intervals. And so the detective story that we kind of told ourselves was, as we were wearing our what scientist hats, we were asking questions like, well, there must be a way to describe these interspike interval distributions. What could that be? What would it look like? And so on. And so in this investigation, we learned how to load the spike data set to figure out its structure. We learned how to plot and visualize the spiking activity across the populations of neurons. And we learned to compute the distribution of interspike intervals for a single neuron. And then we tried out a few functions to fit them to the data to see which one described the data the best. So in a nutshell, this model asked, what is the best description for the behavior of this particular system? And the answer was that a specific model fit better than a specific other one. So the exponential distribution was the best fit to the data. So I wanted to share with you that I also use what models in my own research to give you a broader perspective. So I wanted to tell you about a paper that I actually published quite a long time ago now. This was 7 years ago when I was a grad student. And in this paper, I was interested in understanding the natural statistics of objects around us, the distribution of how big they are, how much they weigh, their density, that kind of thing. So like the size and weight of the stuff around you that's in your office or your home office right now. So your computer mouse, your water bottle, an apple, that kind of thing, things that you typically interact with in the world. And the reason that I was interested in this is because it's been shown that in another context, the natural statistics of our environment, so the distributions of other stuff like color and brightness and luminance, orientations, stuff like that in the world, that all of these natural statistics actually shape our perceptions, which is how we subjectively experience the world. So because we pick up stuff and we manipulate these things all the time, I wanted to know if our experience of how heavy an object is might relate to the distribution of the size of objects and their weight and their density in the world around us. So to do this, I had to first measure that natural statistic. I had to go out and measure the size and density and weight of a bunch of stuff in the world. And then I ended up relating this to how heavy people feel objects to be. So with my collaborators, we wrote some 3D scanning code. We calculated the volume of a bunch of different stuff in the world. We calculated how much they weigh, and then that was enough to give us the density of these objects. And then I also ran a separate behavioral experiment where I asked people to pick stuff up and rate how heavy they thought that it was. And so the what question that I wanted to answer is, what is the relationship between an object size and its density in the real world? And does that fit with how heavy people feel that stuff is? And so what we found here is that a log linear relationship was the best description of the relationship between volume and density in the world. So what this means really is that bigger objects are less dense, and this isn't really surprising, that smaller objects are heavier for their size than larger objects. And this really makes sense because if you want to pick up something, the bigger that it gets, if it gets too heavy because it's the same density as a super tiny object, then you're not going to be able to pick it up anymore. And so the objects that we typically pick up and manipulate in the world have this inverse relationship between volume and density. So what was really interesting, though, is that people seemed to have learned these natural statistics. So when we asked them how heavy they thought the thing was, their answers were like bang on the exact average for how heavy a thing should be if it was that big. And so it showed that they kind of had access to the natural statistics relating volume and weight, therefore density in the world. So I thought that was pretty interesting. And so this was just another example of a descriptive model in neuroscience, that I could describe the relationship between volume and density in the world, and then I could describe how people's answers fit onto that relationship. So this is one example, but there are a lot of others too. So in addition to this natural statistic kind of approach, you could also describe neural firing rates like we did today. You could also describe Fano factors. You could also describe reaction times in a behavioral experiment. You could describe cosine tuning functions for neural tuning preferences. You could describe how the bold fMRI signal behaves under different conditions using a general linear model. There are tons of options here for the what models. And so these are just a few to get your wheels kind of turning. Okay, so the next model that we talked about today was a how model, or a mechanistic model. And so in this one, we examined how different details of the balance of excitation and inhibition make the simulated neural system behave like the real neuron. So wearing our how scientist hats, here's the detective story that we kind of told today. So we first thought about how interspike interval distributions look exponential. That's what we found previously. And we thought, well, there's probably a specific mechanism that makes them look like that. And so then we said, okay, well, what is that mechanism? How would that work in a biophysical system? What bits of the physiology could make it do that? So we know that neurons have capacitance, for example, so they should integrate these signals. And we wanted to see, does that explain this interspike interval histogram? And so we found out, that nope, it didn't work. It wasn't just excitation. It couldn't just be capacitance. We also needed to add inhibition, and we needed to add inhibition in a very specific way in order to get it to be balanced and produce that exponential spiking rate pattern. So to remind you of our learning objectives, what we did through this kind of detective process was we coded the simple leaky, integrate, and fire model of a neuron. And we discovered that it didn't really work properly to produce those spiking intervals that we saw right out of the box. We had to play around and add inhibition in a particular way. We had to add these specific biophysical details in a very constrained format in order to get it right. And so what this meant is that through this tutorial, we got a window into how these details, this balance of excitation and inhibition and these other biophysical details, how they mechanistically bring about the specifics of the system that we're studying. And what we discovered is that the way to get the system to behave correctly is that we have to make inhibition and excitation totally balanced within the system. So that was the tutorial that we did today. And I wanted to tell you that I also use how models in my own research, these how models here. So I wanted to give you a little taste of some of this diversity as well. So as I mentioned back in the welcome message, my own research generally studies perception and metacognition and consciousness. And so the details here are not important. I just want to share with you a little bit of what's going on here. One of the details that we found when we study confidence in this perceptual decision-making kind of way, so that confidence, by the way, is when you see something, how confident you feel that you got it right, that you are able to understand the visual information coming to you or the auditory information coming to you and so on. So when your brain decides how confident it feels in a perceptual decision, like what you think you can see out there in the world, the brain seems to experience some kind of confirmation bias. And so what this means, this confirmation bias here, is that once you've decided that you see a cat and not a dog, your confidence seems largely driven by the amount of cat evidence in the image that you're looking at. Never mind how much it also looked like a dog before you made your decision. So this is kind of a weird thing. Your brain seems to seek information that confirms its own perspective and ignores information that tells it might be wrong. And this is weird because what the brain should do all the time is that you should feel confident when you're likely to be right. Not just when the cat you see is super bright or easy to see, and that information telling you that you are wrong should be integrated into this sense of confidence. Again, please don't worry about the details here. The point is just that the big picture of what kinds of questions we might ask with this observation and this how model, just to show you kind of how I use it. So in my research, I wanted to know how the brain, this squishy wetware in your head, might actually implement this confirmation bias. So what mechanistic details would need to be in place in the system? How would different elements in the perceptual decision-making circuitry need to talk to each other to make this happen? Again, details are not important, but basically we took some well-known properties of the nervous system. So we looked at lateral inhibition, divisive normalization, some kind of classic neural network approaches, but we put them together in a new way. We tweaked them, and we fiddled with how they would mechanistically drive the perceptual decisions, like the decision that it's a cat and not a dog, versus the metacognition or the confidence judgments. And so with the tweaks that we've added to this kind of classical system, we've worked out a potential mechanism for how this confirmation bias might exist in the brain. And it's actually working so far to describe some really kind of surprising behaviors. So this is one of mine, again, but there are lots of others. So the famous Hodgkin-Huxley model, for example. This elegantly describes the morphological structure and the dynamics of how a neuron actually functions. This would be a really great example of a mechanistic model. Okay, and so finally today, we saw a why model, or a normative interpretive model, an ideal observer kind of approach. In this tutorial, we asked why neurons spike in the pattern the way they do, why the exponential distributed spiking rates is the best description for their behavior. So wearing our why scientist hats, we pulled together the observations from the last tutorials and said, okay, we know that the interspike interval histogram looks like an exponential. We know that how that works in the neurons themselves with this balance of excitation and inhibition. But why does the system do that? Why did it come up with the exponential distribution and not another distribution? Why did it feel the need to make excitation and inhibition be balanced in the system? What's the point? Why did the brain evolve this way? And so to answer these kinds of questions, in this tutorial, we first learned what entropy was, which was a measure of the amount of information in a system. And then we computed the entropy of some toy systems. And then we finally computed the entropy of the spiking activity from the data set that we've been playing with all day. And what we found out was that the Poisson or exponential distributed spiking here ends up maximizing the information that can be transmitted in a neural system. Basically, this is optimal. This is the best way that the system could behave mathematically. And so this is maybe why evolution chose this way to do it in essence. So this normative model kind of gives us a window into what factors may have been optimized in one way or another by evolutionary pressures or potentially other forces. So the why models are also a really, really big part of my own research program. So I wanted to tell you a little bit about that. So in a more recent study now, I was interested in using a why model to investigate something that's kind of related to that perceptual confirmation bias and confidence that I just told you about. Only this phenomenon is like really a lot weirder to me. So there's this technique called transcranial magnetic stimulation, which uses a very powerful electromagnet to basically zap you in a particular spot in your brain. And the consequence is that it can disrupt or suppress the neuron's activity in that particular zapped spot. So a couple years ago, actually about 10 years ago now, some researchers had shown that if you use this TMS, this transcranial magnetic stimulation, and you zap people in their primary visual cortex, which is this little, it's the visual hub, and it's that bit of your brain at the back of your head near that bump. If you reach back here, you can feel that bump on the back of your head. So if you zap people there, and if you hold the thing and you zap them there, you can really make them terrible at visual tasks. And this is because the zapping disrupts the visual processing in that region of the brain. But despite the fact that the zapping makes them terrible at visual tasks, they feel even more confident. So basically, you can make them not be able to see reliably, but they think they're doing great. You turn off their ability to see, and they feel like they're doing awesome. So this is kind of weird. And we wanted to see if we could figure out why this happens. And so to do this, we developed two possible models that could potentially answer that why question. These were developed based on previous research. The first model was this Bayesian ideal observer. This is a normative model, which used all of the available evidence it could to access, to optimally estimate the likelihood that its perceptual decision was correct. And the second model was almost exactly the same. There was one important difference in that we didn't allow it to estimate its own internal noise, which is the stochasticity of its own internal neural representations and the noise in its neurons. It just had to guess based on what we called previous experience. So I don't want you to get bogged down in the details here. Again, the details are not the point. The point is we had this question, which is, why does TMS, to visual cortex, make you terrible at a task, but make you feel more confident? And here are two possible answers to that question. One that's allowed to estimate kind of its own internal noise in its system, and another one that is not allowed to estimate its own internal noise in its system. And we wanted to see which one of these fit the human behavior better in our experiments. So we're going to also do this one in a little bit more detail tomorrow. So hold that thought, put a pin in it, and we'll come back to this tomorrow. So just for today, though, what I want you to know is that we wanted to see whether this model or this model was the better one at reproducing the human's behavior. And so then whichever one came out ahead, this would answer this why question. This would give us a clue as to why the people felt more confident when we zapped them, even if they became terrible at the visual task that we were asking them to do. So this is just another example. There are lots of other examples of really fun and exciting why models. Why does the motor system behave the way it does? Why do we see visual illusions? Why does reinforcement work the way that it does? These kinds of things. And so investigating these questions will all require the sort of why or interpretive modeling that you guys played around with today. Okay, I'm going to take off my hat now. So to recap, this was a recap of the what, how, and why kinds of modeling questions and some introductions to how it might be used in other lines of computational neuroscience research. But that's not the only kind of distinction that might potentially be useful. So it's not just about what, how, and why. There's also this other thing that David Marr came up with quite a long time ago now, which has come to be known as Marr's 3 levels of analysis. So some of these tend to map on to the what, how, and why distinction kind of nicely, but sometimes they don't. And so since Marr has been really influential in how we think about computational models in neuroscience, we thought we'd remind you about Marr now if you've heard of him before and his 3 levels of analysis. And for those of you who haven't heard of this before, this is really an important framework to kind of lead into the rest of the school. So the what models of that what, how, and why kind, they're kind of outside this hierarchy. But the why and the how models span Marr's 3 levels of analysis quite nicely. So as I said, the what is outside the hierarchy. The first level that Marr came up with is called the computational level. And so the idea here is that you are trying to describe the objective of the system, which is essentially, why does the system behave the way it does, or why is the system organized in the way that it does? So these are those interpretation why kinds of questions. How close is the system to optimal, and why, and so on. And then it gets a little bit muddier in terms of the mapping between what, how, and why and Marr's 3 levels. But at Marr's algorithmic level, which is his level 2, we start to ask about the data structures and the approximations that are involved, what the runtime of the system might be like to accomplish the various computations and approximations that the brain is trying to actually do. And then, so this is kind of spanning the why model, and then maybe a little bit of the how model. And then we get into Marr's level 3, which is the implementation level. And this is really much closer mapped to the how model of the how, what, and why that we talked about before. And so the idea here is that we're asking about the hardware, essentially? Is it neurons? Is it synapses? Is it molecules? Is it something else entirely? So there's a little bit of disagreement about how exactly the what, how, and why models kind of map onto Marr, but that's okay. As I said, not every model fits nicely into each of those boxes, and so this is just another way to think about it to get you going on trying to understand the types of questions that you want to ask and answer with the models that you're going to learn about over the next 3 weeks. Okay, so the next thing that I want to do, given that there's this diversity of models, what, how, why, and we've got also now Marr's 3 levels of analysis to think about, which is the computation, the algorithmic, and the implementation level. The question then becomes, okay, so how in the world do we choose which models are the right ones? And then we have to think about how to implement them. And so how to choose the right model and how to choose how to implement the right model, this is really a decision process. So we have to think about what questions that we're asking, what goals we want to set for ourselves in the models that we pick. So do we care about what is going on? Do we care about how it is happening? Do we care about why it is happening? Do we want to study the system at the level of cells and synapses, or maybe at the level of information like we did before, maybe somewhere in between? So the introduction that you guys did today is really only the first step. This is just about kind of framing the question. Then you have to implement the model and test it and publish it and do some more experiments and come up with new hypotheses, and the whole process kind of starts again. So this whole exercise really makes modeling a series of decisions. Decision process is what we're calling it, about what we think is important, what questions we want to ask and answer, what toolkits we think are the best ones for the job, how to evaluate our model's behavior, how to interpret your results, how to write them up so that you can share them with the broader scientific community. So in the second part of today, what I wanted to do is to start walking you through this cycle with one of my own papers. This will give you an idea of what this looks like in a real research setting. And we'll start talking about this now, and then just to introduce it, and then tomorrow morning we're going to come back to it in a lot more detail, okay? So as I'm going through this brief introduction, I want you to think about how each of these steps might be applied to the models we talked about today and to the types of questions you think about in your own research. Okay, so I'm going to use as an example this paper that I published about 5 years ago now, because it shows the process kind of nicely. But as I walk through this again, think about how these steps go with your research about the questions that you think are interesting, about how they might be applied to neurons or fMRI data or other types of data sets. Okay, so first we have to frame the question. All right, so in this project that I'm going to tell you about and I'm going to introduce today, the question really came from this observation of the phenomenon called blind sight. This happens when someone has damage to their primary visual cortex. Again, that's at the back of your head. And instead of going blind, they go like kind of part blind. They're still able to use information to avoid obstacles when they're walking, but they really feel like they can't see. So this is kind of the opposite of the thing that I talked about earlier. And so the reason this is interesting is that it kind of separates the seeing from the feeling of seeing. So I'm interested in it from a theoretical reason. But it's also really interesting because just like I told you earlier, if I zap you at the back of your head, I can make you feel more confident, but make you terrible at using visual information. But then there's also been some other papers published that show that the opposite can occur. So this type of blind sight effect, which is unconscious vision, this can also happen if I TMS you in the back of the head. I can make you less confident, but still be able to use visual information. So there's this dichotomy here, this controversy, and we wanted to get into that a little bit. So we had to develop a set of models which would compare what's going on. And so those are the blind and aware models that I told you about just a couple slides ago. So then we had to develop so those are the models. Then we had to develop a behavioral experiment that would control for all sorts of other things that we didn't want contaminating the effect, and develop a TMS protocol based on previous findings that would hopefully induce the effect that we were hoping to study. So we had to learn how to zap people in the right spot in order to induce these types of effects. Then we had to do the behavioral experiment. And then we had to fit the model to the types of data that we got. And then we had to evaluate the 2 models that I told you about against the behavioral data to see which one was going to win, which one was going to fit the data the best. And so finally then, when we figured out which model fit the data better, and I'll tell you about this a little bit more tomorrow morning, we were in a position to answer that why question. So why does TMS appear to induce blindsight in some cases, but not in other cases? And specifically, why does TMS actually make you feel more confident, or how could TMS really make you feel more confident in your vision, even as it really messes up your ability to see? And so the answer appears to be that people are not optimal in their confidence judgments after all. This was a big surprise to us and maybe to others in the field. And so basically this means that people can't make use of all the information available to their visual system. And the result seems to be this funny behavior where you think you're doing great, but you're actually really just terrible at the visual task. So all of these steps are going to come back in the tutorials over the next several days, and also throughout the Neuromatch Academy course. So the same logic can be applied whatever it is that you're studying. This is just one way to do it, one of my examples. And this can go all the way from the simple spiking neurons that you had in today's tutorials, all the way through some of the super complicated and really exciting questions that we're going to tackle over the next few weeks. So, okay, the last thing that I want to do today is take kind of a zoomed out, bigger picture look at the diversity of models that are available. This is kind of the point of Neuromatch Academy, to learn about the big diversity and a plethora of models that are out there. And this means that you can now start to learn how to pick which model goes best with the questions that you want to answer. So it's not just about the scale of abstraction, from neurons to systems to people, and it's not just about the type of question, the what, the how, or the why. Paying attention to these features is great, but it's also important because they help you define the goals of a particular modeling framework, which can really help you define how to evaluate which model is the right tool for the job, and given a model, whether it was successful or not in producing answers to the questions that you really wanted to ask. So knowing which models are out there and how they differ, this also helps us think about what we can expect from a model, both in terms of what it can do here, what we should expect from the model here, but also defining its limitations, what it can't do. And so the point here that I want to get into in these last just couple minutes is that everybody has their own objectives, their own goals. And the important thing to note here is that all of these potential goals of various models at the different levels of abstraction, they answer and ask different questions. All of them are valid and useful. And so the last thing that we're going to do today, just in the last couple minutes, is to take a look at the goals aspect here. So what is it that you want your model to do? What do you want to get out of it? What can you expect it to accomplish? So each goal is not necessarily better than any other one. I wanted to get you started then with some possible goals to think about when selecting your modeling framework and approach. So as I go through these, I'm going to put a lot of text on the slide here. I want you to start to think about which ones you think are important. And remember, there's no right or wrong answer here. So the first potential goal is you want your model to be useful. You want your model of the nervous system maybe to be good at solving a real-world problem. For example, a model of the visual system might be able to solve a challenging problem in computer vision or something like that. And so you could also want your model to be normative. Some models provide the optimal solutions to problems that exist out there in the real world. So they're used maybe in domains where behavior or neural properties are expected to be optimal or near optimal. You also might want your model to be clinically relevant, to produce insight that's relevant for developing or evaluating a clinical intervention of some kind. You also might want your model to inspire experiments, to change the way we think about a problem and raise interesting new hypotheses. You also maybe want your model to have microscopic realism. You want to describe microscopic properties of the brain, like synaptic or pharmacological or cellular level properties. Or you might care about macroscopic realism, to describe properties of brain areas and networks instead. You also might want behavioral realism. So there are some models that can faithfully describe and explain behavioral phenomena. Is this one of the types of things that you're interested in doing? You also might want your model to be representational. Some models aim to use representations of information that might be similar to the representations that are used in the brain. You also might want your model to be compact, to be succinctly expressible in math or in code. Maybe this makes it easier for humans to understand these models. You could instead want your model to be analytically tractable. Some models might be understandable through math equations instead of numerical simulations, and so maybe this is very attractive to you. Couple left, you might want your model to be interpretable. Some models can be easily interpreted with respect to how they work, and maybe this is really important for you to understand how the brain might implement computations that you care about. And then the last one that I'll put up on the slide here is beauty. So maybe some models are symmetrical or balanced, or they resonate well with the way that we think, and this is also a valid modeling goal. So you might be thinking, okay, there's a ton of goals that I just put up on the screen, and then we've got this what, how, why thing, and then we've got this Marr's 3 levels of analysis thing. Where do I start? Should I start maybe with the what models and then move on to a how model and then move on to a why model for a particular problem? So it's not really a fair thing for me to ask you, though, to say, like, should I start here and then move here and then move here? Because there really is no right answer. There's no right kind of model to build. There's no right kind of goal. There's no right kind of level of analysis. The type of model that you want to build depends entirely on your goals, on what kinds of questions you want to ask. And what's really interesting here is that the diversity of goals and models available means that we really have a lot of choices to make. Everybody has a different idea of what makes a good model. Different models ask and allow us to answer different questions. And so this is why model diversity is really good. So as a final thing that I'll put up here, this is a graph of a survey that Conrad Cording and Gunnar Blom and Paul Schrader and Kendrick Kay did just a little while ago. And in this survey, they asked scientists which goals they thought were important. And what I want you to pay attention to here is not the agreement, but the disagreement, the size of these error bars. There's a huge amount of variability here. People think differently about which types of models are the best ones, and that's okay. So the last thing that we'll do today, as opposed to all the other days, is we'll launch into the Q&A period by starting to talk everybody, all of you, about which of these goals you like, which of these guys you think are important, and maybe why. And then we'll see you back here tomorrow morning to launch into how to actually really get our hands dirty with that decision process and how to use it to our advantage. So this is kind of the whole point of Neuromatch Academy, to help you see that diversity of model goals and options and variants and choices. This whole program where we're doing all of this stuff is so you can learn how to make really good choices about how to approach really interesting questions about your data and others' data. So the diversity is the point. And so now we're going to have this wonderful discussion about what kinds of models you think you might want to use. Not just about the models themselves, though, but about the goals that they're going to help you accomplish. So here are those goals to get you started, and I hope you're going to have a lot of fun. See you guys soon.